Good morning to Madam Nat and my fellow friends. Today I'm going to tell about my topic, which is about Human Genome Project. Okay, Human Genome Project. What is Human Genome Project? It is an international research program which is to determine the sequence of our human genome and identify the gene. Okay, it's coordinated by the National Institute of Health, which is the synchronized name is NIH, and US Department of Energy. Okay, the beginning for this research is to understand the blueprint of human body. Reason why Human Genome Project is developed is one is to understand genetic changes that cause cancer, and US needed efficient way to study and long-term DNA damage by radiation. Okay, important of Human Genome Project. One is identify the location of human genes. Second is create tools to analyze the data. Third is the sequence numerous genomes of other organisms like brevier yeast, the fruit fly, and roundworm. Okay, the study is study the similarity and difference between human genes and other organisms. Besides that, it's discover function of particular genes, and it's identify genes that crucial for the life. Okay, there are some methods which is DNA sequencing method. One is Maxim Gilbert chemical degradation. Second is Sanger sequencing. Third is Price sequencing. And fourth is Illumina sequencing. And fifth is solid sequencing. Okay, that another dystrophin. That is the biggest known human gene, which is made up of about 2.4 million bases, and it contains 79 exons. It took 16 hours to transcribe and split up introns. It's involved in signaling events and synaptic transmission. It's also discovered using it's by this dystrophin. It's discovered using the Human Genome Project.
Okay, ladies and gentlemen, we have talked so much on the details of dystrophin, and now we'll look into the actual structure of dystrophin itself. So this is the structure of dystrophin. And next, we will look into the junk DNA. As we all know, half of human genome are made up of non-coding DNA. Non-coding DNA is actually seem useless, not like the coding DNA. So it is called as junk DNA because it only seems useless, as useless as the rubbish is. But nowadays, those non-coding, the seem useless DNA play an important role in controlling genes activity. How does it control gene activity? Well, it codes for genetic switches in body. And we'll look into the complete Human Genome Project. Well, Human Genome Project completed its first draft of Human Genome in 2002, where almost 20% of code had yet to be sequenced. However, during 2003, a new version of Human Genome Project is released. And then the scientists estimated about 100,000 genes in human. Nevertheless, the results show only 20,000 genes from the estimation. So we'll look into the Human Genome Project breakthrough. What is the benefit of Human Genome Project development? The first one is it enables researchers to make discoveries more than past times. And next, it becomes a valuable new tool for studying human origins and history of species migration. Or in the other words, the Human Genome Project can help us to deny the statement that we are the descendants of the monkeys, just like in the Darwin's theory. So the third one is scientists could look at diseases association at genome-wide level. And next is found out about 98.5% of DNA has nothing to do with genes. It's just useless. And the last one is the creation of newer, faster and cheaper methods of gene sequencing through the development of Human Genome Project. Lastly, we will look into the prediction on Human Genome Project. In the future, the scientists may pinpoint genome that make us Homo sapiens, and we can hope that gene therapy will cure diseases. The third one is the actual definition of junk DNA will be discovered. Next, everyone's genomes can be sequenced and prevent diseases. And the last one is possibility on seeing the movement of genomes and their actions. So we can see that the Human Genome Projects provide us with many benefits. So that's all from us. Thank you.